Right, my dear students. So when we expose these plantlets from test tube to environmental condition by a process that is a hardening, right? This is a way by which we can develop a plant, right? Or plant organ or root or stem or leaf or any other portion, that is called an organ culture. The next type is meristematic culture. Meristematic. Second type is meristematic. What is a meristematic culture? Simply, meristematic culture is a culture of a meristematic tissue or a meristematic region without fusion of male and female gamete. Actually, it is an artificial method by which we can develop virus-free plants, right? Virus-free plants are produced or cultured by a technique that is called the meristematic culture. Actually, meristematic culture in which simply a growing point of, the shoot is taken. What is taken here, my dear student? Please understand. Shoot. This is a stem portion and that bears number of leaves, right? Here you find the leaves are there. And when we cut here, we can get a piece of land. With this is a growing portion, right? After cutting, we can get the growth of the meristematic region or a growing point of the stem, right? It is to be shown as it is. So this is a meristematic region after removal of or the meristem. Meristem is nothing but an apical portion, which is nothing but the growing point of the stem, that is called the meristematic region. So when we cut the plant into, when we get a piece of plant, right, after removal of the large size leaves are enclosed inside of the stem, we can get this growing point of the stem as it is, right? Again, when it is transferred to culture medium, what will happen is it also initiates to form a number of leaves, right? This is a growing portion of the stem, right? And these growing portion, again, it is to be removed. The large size leaves are also originated. So totally, the growth of the leaves from the meristematic region or the growing point of the stem, completely the leaves are removed. After removal of these leaves, these tissues are transferred. These tissues are transferred into test tube containing the growth substances, right? When we transfer this meristematic tissue in test tube containing cotton wig, right? Here you find it. Cotton wig is there, right? So simply these tissues are transferred. After some times, what will happen is the next stage, let us find the growth of these callus. Simply I can say it is a callus. Already I said callus is an unorganized mass of tissue. This callus tissue is differentiated into, on one side you find shoot system, and another side we can find the growth of the initiation of root system, right, my dear student? So as a result, we can get the plant the small plantlets are originated inside the small plantlet. The small plantlets are also exposed or to the environmental condition, right? By a stepwise manner, by the process called the hardening process. So finally, the plantlets are originated from the test tube, right? We can get entire plant, right? So this technique is also known as simply meristematic culture. So by the process that is called the hardening process. Hardening. Hardening is the process of exposing plantlet from test tube into environmental condition. I mean soil. The transfer of plantlets which are growing inside of the test tube, which is a transfer into soil, that is called the hardening process. Already we have discussed about there are several steps are involved in plant tissue culture, right? Culture medium, culture vessel, hardening, sterilization. So many techniques are there. So many steps are also involved. So the final step of plant tissue culture is the hardening, exposing a plantlets from the test tube into environmental conditions. So this is a way by which we can develop meristematic tissue or meristematic culture, right? So simply meristematic culture is nothing but the grain point of shoot is cut into piece and the piece are transferred into culture medium, and after transferring what will happen is growth of the culture, growth of the transferred tissue into test tube simply that will form a callus. Callus is nothing but it is an unorganized mass of tissue. So when it is a transfer into test tube containing nutrient or growth medium, which is covered with help of cotton wing, from the cotton wing again it is a transfer to another medium, which is incubated for a few hours, right? After some time, what will happen is there is a initiation of a root shoot in test tube containing nutrient or growth substances. So finally, the plantlets are nothing but it's a small nursery plant which are initiated from the callus region. So from the test tube, the plantlets are exposed or transferred to the environmental condition in a stepwise manner, not directly, right? So by the process that is called the hardening. So this is simply something about meristematic culture. So next we pass on to protoplast culture. That is very interesting topic, my dear student. Please understand. The next topic is protoplast. Protoplast culture. Protoplast culture is a kind of a plant tissue culture in which protoplasts are isolated, which are fused and cultured, by which simply a somatic hybrid is developed from a technique that is called the protoplast culture. Dear students, you should understand what are protoplasts. Generally, protoplasts are nothing but, now this is a cell, right? The cell always contains at the center you find this is a nucleus, right? It is a nucleus. And just around the nucleus you find cytoplasm. What is it? Cytoplasm. And around in the cytoplasm you can find a number of cell organelles, right? Like mitochondria, Golgi apparatus, etc. But the cell is surrounded by a plasma membrane. The green portion is called simply plasma membrane or cell membrane. Plasma membrane. The plasma membrane which is not bound by cell wall. Here cell wall must be bound. Right here without cell wall. This is a cell wall. The cell wall is not present, which is not bound by plasma membrane. That is called the protoplasm. So what is protoplast? Protoplast are the cell without cell wall, but it is bound by plasma membrane. That means the green portion indicates plasma membrane. So simply I can say it's a nagged cell, but that contains cytoplasm and nucleus, right? So that is the difference between protoplast and this cell. Now what we are going to do in protoplast culture. Actually, by using a protoplast, we are going to make new plant. Entire plant is developed from the protoplast. So that is called protoplast culture. So the answer is hidden in the title. Protoplast simply the protoplast are developed and fused and the culture and to be made a new plant. The development of a new plant from the protoplast, right? That is called the protoplast fusion, protoplast culture, or protoplasmic fusion, or somatic hybridization. There are several names of the instead of protoplast culture, right? What are they? Protoplast culture. It is also known as, simply I can say, it is also known as protoplasmic fusion. Protoplasmic fusion, or it is also called as somatic hybridization. It is also called as somatic hybridization, because the somatic cells are involved in the fusion of protoplast. As a result, a hybrid plant is developed by a technique that is called the protoplast culture or protoplasmic fusion, or it is also called as somatic hybridization. So simply the development of an entire plant by using of protoplast, right? So that is called the protoplast culture. Okay, the protoplast culture, it consists of some steps, right? There are some steps are involved in protoplasmic fusion or protoplast culture or somatic hybridization. So what are the steps are involved, right? Let me find out one by one. Dear students, please don't get confused, right? From organ culture, from meristematic culture, from cell suspension culture, right? So actually, the protoplast culture, it consists of following steps. The first step is isolation. The second step is fusion. And third step is culture. Then fourth step is Selection of somatic hybrid. Selection of somatic hybrid. Yes, students, please understand. My dear students, did you understand? Students, are we audible?
Yes, sir.
In protoplasmic culture or protoplastic culture or somatic hybridization, how many steps are involved? There are
4 steps.
very good. What are the 4 steps? The first one is
Isolation, fusion, culture, selection of somatic hybrids.
very good, ma'am. So there are only 4 steps are involved. Okay, let me take the first step. What is it? Isolation. Actually, what is isolation? Isolation is nothing but the protoplasts are isolated by using of some chemical agent, right? So that is called isolation, in which some enzymes are involved, some chemical agents are also involved. So I'd like to mention the nature of a chemical compound, simply 0.5%. That is a macrozyme is taken. Macrozyme. Here in your textbook, there is a mistake. There is a macrozyme. That is an error. Please understand, it is a macrozyme. It's a complex mixture of chemical substances. In addition to that, 2% of simply cellulase. 2% of cellulase is taken in order to isolate protoplasts from the leafy portion, which are collected from the photoplant when we use as a explant, right? And in addition to that, 13% of sorbitol or mannitol is added in a petri plate. Simply, what is taken finally? 13% of sorbitol, or it is also known as mannitol. Then, in addition to that, one more chemical agent is added, that is a 20% of, right? 20% of sucrose is also involved in first step. What is the first step? Isolation. In order to isolate, what are the chemicals are applied? First thing is 0.5% of macrozyme is taken, which is mixed with 2% of cellulase. It is an enzyme which is used to remove a cell wall in the explant portion. Then, in order to maintain the viability of the cell, right, the cells are kept in beaker containing or petri containing 13% of sorbitol or mannitol. So finally, the isolated protoplast are transferred into another petri plate containing 20% of sucrose. Why it is transferred in 20% of sucrose? Because in order to retain its viability of the protoplast, right? So these are the chemical components or involved in the first step of protoplast, that is isolation, right? Sometimes from the isolation, you could expect some question. Dash percentage of cellulase is taken in isolation of protoplast. That is the question. If it is asked, what is your option? Dear students?
2%.
Very good. And dash percentage of sorbitol is added in isolation of protoplast. If it is asked, what are going to answer? Dear students?
13%.
13%.
Very good. We mentioned the ratio of macrozyme, which is involved in isolation. One of you, please.
0.5%.
Very good. 0.5%. Then in addition to that, these chemical substances are maintained in a petri containing mixture of chemical compound. Its pH value is right 5.4. So will you mention the pH value which is maintained in petri containing chemical compound? Could you mention please?
5.4.
Very good. 5.4. So what I have given here in isolation of protoplast, definitely you can expect, right? So this is a step there, which is taken, right, as a first step. Then second step, what is the second step? Fusion. Actually, isolated protoplasts are fused or joined together by an agent that is called a fusogenic agent or fusogen. Please understand, dear student, one of the chemical agent that is called a PEG. That means polyethylene glycol. P stands for poly, E stands for polyethylene, and G stands for polyethylene glycol. It is nothing but it is a fusogen. Polyethylene glycol is nothing but it is a fusogen. What is fusogen? Fusogen is a chemical. Fusogen. Actually, it is having ability to fuse two different protoplasts. That is called a fusogen. Actually, this is a fusogenic agent, namely polyethylene glycol, which is going to fuse two different protoplasts, which are involved in protoplasmic fusion or protoplast culture, right? So that is called a fusogen. Along with the fusogen, right, in addition to that, one of the nutrients also plays an important role in order to retain its viability of the protoplast. That is called simply calcium. The calcium, which also plays a prominent role in the formation of callus, right? So when we use polyethylene glycol as a fusogenic agent, right, calcium is also added. So these are the steps which are involved in fusion, right? In addition to that, it is also incubator. It is also incubated by the temperature simply 25 degrees Celsius. So it requires the optimum temperature of protoplasm fusion is at 25 degrees Celsius, right? So that is the nature of the fusion. Second step. I'm talking about second step, fusion. Dear students, could you mention the name of the fusogenic agent which is involved in protoplasmic fusion?
Polyethylene glycol.
Glycol, very good. What is it? It is a
Fusogenic agent.
Very good. Sometimes you can expect the question in 2 marks or in 1 mark question also. Both and B. Which of the following is known as a fusogenic agent? A. Polyethylene glycol. B. Glyceric acid. C. Glycolic acid. D. All the above. Right? If it is asked what you are going to take and give us answer.
Polyethylene glycol.
It is a fusogenic agent. Sometimes it may be asked as a 2 mark question. What is PEG? PEG is nothing but polyethylene glycol. It is known, it is considered to be as a fusogenic agent because it is having ability to fuse two different protoplasts, right, which are involved in protoplasmic fusion or protoplast culture or somatic hybridization, right? And so hence it is known as a fusogenic agent, right? So that is very important area to be concentrated, to be focused in examination point of view, right? Please understand. Okay. And next step is a culture. Actually, I'm talking about third step is the culture. Protoplast are isolated in first step, and the isolated protoplasts are fused or joined together by using an agent that is a polyethylene glycol, and they fused two different protoplasts or culture are developed or copying into number of copings or made into number of copings or produce a number of cells, number of protoplasts, right? So that is called culture. In culture is a technique in which simply the isolated protoplasms are fused by using polyethylene glycol, which is to be developed, right, in a culture medium. So here, in order to culture or in order to develop, right, it requires particular intensity of light. So the light is, I think, 1000 to 2000 lux, the unit of intensity of light, right? The unit of lux is nothing but a unit of intensity of light is called as lux. So what is a unit? 1000 to 2000 unit, right? Intensity of light. It is exposed. So the intensity of unit of light is measured by lux, right? Nothing in it. So finally, it is made as a plating technique, right? The cultured protoplast, or simply I can say it is developed into the plating techniques. So finally, this is a hybrid, two different protoplasts are now. This is a protoplast. When we use the protoplast, right, we can get, right, that is a hybrid, the fusion of only cell, fusion of only cytoplasm, not nucleus, right? So that is called, simply I can say, hybrid. So what is hybrid? I am going to show you later. Hybrid is a fusion of cytoplasm without the fusion of nucleus. That is called a hybrid. Now the hybrid is followed by the fusion of nuclear fusion. Hybrid is nothing but it is a fusion of only cytoplasm, not nuclear fusion. That is called the hybrid. The hybrid is followed by fusion of nucleus, that is called a hybrid. Such a hybrid is called a somatic hybrid. The somatic hybrid is nothing but it is a process of producing somatic hybrid from the somatic cells, that is called a somatic hybridization. A hybrid is derived from two different protoplasts which are fused to form a hybrid. Then hybrid is developed into hybrid. This hybrid is developed into hybridization. So simply I can say it is a process of producing somatic hybridization, or somatic hybrid is called the somatic hybridization. Actually, somatic hybrid is nothing but a hybrid is obtained from hybrid. A hybrid is fused by two different protoplasts, which is followed by nuclear fusion, that is called the somatic hybrid. So this process of producing somatic hybrid is called the somatic hybridization. Here, somatic hybridization. Somatic hybridization, right? So please, it is very interesting area, somatic hybridization. Okay, let us see what is a somatic hybridization. I am going to explain the flow chart that I have collected, that you can understand easily, right? Now, this is a somatic hybridization or protoplast culture. So the first picture indicates it is a plant, right? The plant consists of a number of leaves. So each leaf, each part of the plant is known as a explant. What is it called? Explant. No, explant is nothing but the plant part used for plant tissue culture technique is called explant. Now the explant or the leafy portion is sterilized with the help of some chemical agent or alcohol, right? So sodium hypochloride or potassium hypochloride is a sterilization. It is a sterilant which is used to get removal of microbes from the explant. That is called a sterilization. Actually, sterilization is a process which is employed to get removal of microbes from the explant. Now, the sterilized leaves are cut into a number of pieces. From the pieces, in second step, right? We are going to remove, right? The epidermis, which is the outermost layer of the leaf. After removal of epidermis, we can get the peeled segments of leafy portion, right? So after removal of these epidermis, we can get the protoblasts, which are plasmolysed, right? That means the released plasmolysed cells are transferred into the enzyme mixture, as I said, right? The enzyme mixture, it consists of 0.5% of macerozyme and 2% of cellulase, right? You can say cellulase, and 13% of sorbitol, right? Sure, please understand that I have explained to you sorbitol. The sorbitol is also mixture. It's a chemical mixture in which when we transfer the plasmolysed cell into test tube containing mixture or this chemical compound, what will happen is the cell wall of this protoplast is gradually removed. So simply I can say the cell wall, which is digested by the enzyme, that is the cellulase. So cellulase here that is involved in removal of cell wall. So as a result, we can get the naked protoplast, naked cell, that is called the protoplast. The protoblasts are taken and put into test tube containing debris, right? Now these protoblasts which are transferred, which are kept into test tube containing a chemical compound, that is in which simply the water or the sterilant compound or sterilant or liquid medium is added in order to remove microbes or in order to wash, right? So after washing the dust particles from the protoplast are taken out, and the isolated protoplasts which are released from the test tube, the protoplasts are copied or made into number of copies. So by a technique, that is a plating technique. So plating technique is a technique by which the protoplast number can be maximized or copied. So after copying of this protoplast, they may start a regeneration of cell wall. I'm talking about this is a portion where we find the protoplasts they are going to reform, they are going to regenerate their cell wall around the cell, right? Around the protoplast. So as a result, we can get a number of protoplasts, number of cells are regenerated their wall. After formation of cell wall around the protoplast, we can get a group of cells. They are called as clumps of cells. They are called simply clumps of cells. That means a group of cells are initiated. So when we transfer these clumps of cell in beaker containing or petitris containing growth substances, there is an unorganized mass of tissue that is called callus, right? Here callus is originated. The callus, what is callus? It is a group of tissue which is undifferentiated into root and tissue. Such an undifferentiated mass of tissue which is differentiated into root and tissue. And root system which grows towards light, and shoot system which grows towards light, and the root system which grows towards gravitational force, right? So here you find that there are two portions, the green portion and the red portion. The green portion is considered to be as a shoot system, and the red portion considered to be as a root system, right? So after formation of root and the shoot in test tube containing growth substances, it is to be transferred into environmental condition by the process that is a hardening process. What is it? Hardening. So hardening is a process by which exposure of plantlets from test tube to environmental condition. From the environmental condition, the plants are developed into new plant. So this is simply something about protoplast. So simply, ultimately, just I'm going to explain simply. Explant is nothing but a plant pot which is collected from the water plant, which are converted into simply unorganized mass of tissue by several steps, right? And finally, the plantlets are developed into test tube containing growth substances. From the test tube, the plantlets are transferred or exposed to the environmental condition. So, by the process that is called the protoplasmic fusion. Simply, protoplasts are isolated from the explant, which are involved in different stages by removal of cell wall for retaining its viability, and the regeneration of cell wall. After regeneration of cell wall, the protoplasts are converted into clumps of cell, and the clumps of cells are developed into unorganized mass of tissue. That is a callus. Then callus is differentiated into root and is shown in a test tube. From the test tube, the plantlets or nursery plant, which are transferred to environmental condition. By the way, the development of an entire plant from the protoplast. Right? So this is called a protoplasmic fusion or protoplastic culture. Right? So thank you for joining, my dear students. Did you understand? Dear students, almost our time is over. Did you understand today what I taught you? This is very important.
Yes, sir.
Important area.
Yes, sir.
This is very important area. Please concentrate, right? Sometimes you can expect write an essay on protoplasmic culture or protoplast culture or somatic hybridization, right? I'm going to give you home assignment.